Welcome back to our channel. Today, we're diving into another important immunohistochemistry test, the CDX-2 test. This test plays a crucial role in identifying carcinomas of intestinal origin. Let's explore everything you need to know about it. Pre-test information. Before we proceed with the CDX-2 test, it's essential to gather some critical pre-test information. Make sure to provide a copy of the histopathology report, the site of the biopsy, and a detailed clinical history. This information will help guide the pathologist in accurately interpreting the test results. Specification of test. The CDX-2 test is an immunohistochemistry test that specifically detects the CDX-2 protein. CDX-2 is an intestine-specific transcription factor, crucial for regulating both proliferation and differentiation in intestinal epithelial cells. It is an invaluable marker in identifying carcinomas that originate from the intestinal tract. Diseases associated. This test is particularly useful in diagnosing carcinomas of the intestinal origin. CDX2's restricted expression in the small and large intestines makes it a reliable marker for these types of cancers. Specimen used. The specimen used for the CDX2 test typically includes tissue samples obtained from biopsies. These samples are prepared and processed to allow for accurate immunohistochemical staining and analysis. Components included. The CDX2 test involves several key components: the primary antibody that binds specifically to the CDX2 protein, a secondary antibody conjugated to a detection system, and the chromogen that visualizes the staining. These components work together to highlight the presence of CDX2 in the tissue sample. Test report time. The test report time for the CDX2 immunohistochemistry test is typically around 7 days. This timeframe allows for the careful preparation, staining, and analysis of the tissue samples to ensure accurate and reliable results. Test information. CDX2 is an intestine-specific transcription factor that plays a vital role in regulating both proliferation and differentiation in intestinal epithelial cells. Its restricted expression in the small and large intestines makes it an essential marker for identifying carcinomas of intestinal origin. By detecting CDX2, pathologists can more accurately diagnose and differentiate intestinal carcinomas from other types of cancer. That's a wrap on the CDX2 test. Understanding this test and its significance in identifying intestinal carcinomas can greatly aid in accurate diagnosis and treatment planning. If you found this video informative, make sure to like, subscribe, and hit the notification bell for more insights into immunohistochemistry tests.